hopefully that makes sense in terms of cell structure and function. Again, ask any questions. We'll get to it in a little bit. But for now, we're going on to the cell cycle. Again, this is a pretty detailed diagram. Most of the diagrams we've got are relatively detailed, so I suggest there's a bunch of good information, like come back after and look at these slides in a bit more detail. Some of them like even a little bit too detailed, like kinetochore, you don't really have to know that. Generally, I kind of hate this diagram. It's a bit confusing because it goes anti-clockwise. I don't know. I just like it when they go clockwise. But we have this idea of interphase, and then we have mitosis. So these little ones are like prophase, metaphase, anaphase, telophase. Those are steps within mitosis. So this whole thing is M phase, mitosis. What we're looking at here is interphase. So this is before the cell divides. So basically, in general, this eukaryotic cell cycle, it's about growing and then dividing and growing and then dividing. So relatively logical approach if you think about it. So we have our three main phases. So we have G1, we have our S phase, and we have G2. So I'll talk about these in a second as well. But G1 generally, like G, we're thinking of like growth, basically. That's how I used to remember it. So the cell is growing. S phase, the cell is synthesizing. So basically we're just doubling the DNA, because remember, we're a cell. We want to divide. We want to go into two. We have to double our DNA so we have enough DNA for another cell. If we didn't double our DNA, then like if we had 10 pieces of DNA to begin with, then we've got 5. Then we've got 2.5. It doesn't really work that way. So you have to double it to 20. So then you can divide into 10. Double it to 20. Then you can divide into 10. If that makes sense. So yeah, that's the general overview. So that's our interphase when we're working towards mitosis. And mitosis is the actual separation of the cell. Okay, so talking about mitosis, we have our little acronym PMAT. Again, you guys may be on top of this. You may have been taught it before. Remember that interphase is not part of mitosis. It's separate. So here is where we're getting like our growing, stuff like that. So you can see replication. We then move on to our P, so our prophase. And this is where our chromosomes start to become visible, basically. We can see we've got our replicated DNA. They're getting more condensed. They're basically getting like thicker, I guess you could say, because they need to be condensed so that we can, I guess, like move them around the cell a little bit more easy. So that's what we're thinking about in prophase. Then in metaphase, think of M for middle. So we're lining up along the middle of the cell, as we can see here. We have our little spindles. You can think of like spindle fibers, and those will attach to the middle of the chromosomes. So that's what we're thinking about in metaphase. Think about middle. We're lining up. And then that leads on very nicely to our anaphase, where we've lined them up nicely. We've attached our little like harnesses, I guess you can think, and then we're pulling them apart. So we're now separating our chromosomes, because remember, they're replicated. There's 2 of them. So now we're separating them. So one goes to one cell, one goes to the other. So we have the right genetic information in each cell. So we can see our little spindle fibers, they retract. I don't know, think of it like a bungee cord or something. So they're pulling the spindle fibers, and again, we're going to either side. And that makes sense because, again, that nicely leads us. Think about how it's leading. It's a process. It's a flow. So now that we've got them on either side of the cell, that's really great. We can just cut them in the middle, and then we've got 2. So we then have our telophase. So this is when we're now on separate sides of the cell. We now start to become enclosed in our little nuclear membrane. Remember that in general, like we talked about our nucleus before, it's our little house for our DNA. When we have our prophase, our metaphase, that kind of gets broken down, and our chromosomes just go wherever they want to go in the cell. Now we have to put them back in our neat little box. So we can see our nuclear membrane, as you can see here in yellow. This is reforming, so we're getting them popped in, separated nicely. Our chromosomes basically begin to decondense, so they kind of unravel. They don't have to be as tightly packed as they were. So then they basically become invisible again. And then you have interphase. So then each of these individual cells, understand that they are separated by this point. I know they look nice and conjoined, but they are separated. And then you've got your interphase, and that starts again. So then those 2 cells will continue to grow. They'll replicate their DNA, and then they'll divide. So hopefully that makes sense. So yeah, prophase, you just think of kind of the chromosomes appearing, the nuclear membrane disintegrating, I guess you could say, or breaking down. Metaphase, think lining up in the middle. Anaphase, think of apart. Telophase, we're kind of getting that nucleus reformed. Remember, there is an important step of cytokinesis, and that's the actual separation. Cytokinesis is not a part of mitosis. So as you can see, we'll talk about it in a second. But as you can see here, telophase and cytokinesis. So cytokinesis is the actual separation. But yeah, so we think of PMAT as those four major steps of mitosis. So here is cytokinesis. So this is what I'm kind of talking about here. So basically, you don't have to worry too much about terms like cleavage furrow and things like that. But basically, now we've got our two like mini nuclei. We just basically pinch off that middle bit. So then now we're separated into two cells, as you can see here. So it's just kind of what you expect would happen. In plants, it is very similar. However, because there is a cell wall, remember in animal cells, we don't have cell walls. In plant cells, they have cell walls. So that's something that needs to be accounted for as well. So everything is the same. You've got your PMAT that's happening all nicely. To create this cell wall division, you have what is called a cell plate. So that basically begins to grow. And you can see they're basically just these structures that will give rise to this cell plate. And then you've got some other stuff going on. Again, you don't need to know this too in depth, but you just have to be aware that an animal cell doesn't have a cell wall, so you just have to split and you're fine. With cytokinesis in a plant cell, you have that extra step of forming that cell wall. So yeah, you can have a read for that process in a little bit more depth, but it's not too important to know all these kind of keywords and things like that. It's just important to understand the difference, as you can see in these diagrams ultimately. Hopefully that makes sense. And remember, this is kind of separate to mitosis. Like I know there's a little bit of, I don't want to say debate, but like sometimes it's very easy to confuse cytokinesis as part of mitosis. Just understand that it is separate. Okay, so compared to mitosis, the way that prokaryotes divide, or for example, bacteria, binary fission, it's so simple. They literally just replicate their DNA and cut in half. So it's almost like the telophase part of mitosis, because you don't have to account for this like separation of like this intricate separation of your chromosomes and stuff like that, because in our prokaryotes there is one circular chromosome. So as you can see here, kind of this black squiggle represents that chromosome. The blue one is that now replicated. Again, they just go to either side of the cell splits in half, you have two cells. Very simple process. It's not as complex as mitosis, and it happens much, much faster. That's why we think of like when you think of bacteria, like what you guys know about bacteria, they replicate really rapidly, and it's super simple because it's such a quick, efficient process, really. So this can happen really, really rapidly. You're just doubling that genetic material and pinching off in the middle. That's ultimately it. But just be aware, things to be aware of. The idea that you have one circular chromosome. You have plasmids. These are just kind of other little offcuts of DNA, I would say. You talk about it a little bit more in 3-4. And then importantly, you have ribosomes. Remember that ribosomes, the difference between prokaryotes and eukaryotes have membrane-bound organelles. Ribosomes are not bound by a membrane. So that's kind of why they're in prokaryotes. Okay, so that was kind of talking about mitosis, I guess going backwards or technically forwards, like looking at the other parts of the cell. So we have our G1, which you talked about, it's literally just growing. S, you have the chromosomes duplicating. G2, growing a little bit more, preparing for mitosis. G0, this is something else. This is like a resting state. So we'll talk about here. The eukaryotic cell cycle is very complex, as you know. There are a lot of things that can go wrong, and things are generally bound to go wrong sometimes. So it's really important that there are checkpoints, because obviously if you were to have a cell that has uncontrollable growth, or if something's gone wrong with the synthesis of the DNA and that cell continues to divide, there can be other consequences further on. So these checkpoints are really important. So we have our G1 checkpoint. So after growing, it's just checking for these kind of stuff to make sure that nothing's too wrong there. You've then got your DNA synthesis. You then grow a little bit more. we're now at our G2 checkpoint where the cell is checking, okay, did the DNA replication go okay? What is the cell looking like? Is it big enough for division? And then we have a checkpoint in the middle of mitosis as well at metaphase, and that is checking to make sure that those spindle fibers that we talked about, that they are attached properly to the chromosomes. Because again, if you pull too much of a chromosome or not enough of a chromosome, you can get that kind of imbalance in genetic information in your cells. So hopefully that makes sense. And if there were anything to go wrong at these checkpoints, the first kind of call of action is repair it. So if you have DNA damage or something like that, the cell will try to repair it. It'll repair it and then it will go on with the cell cycle. If the damage is irreparable, the cell will basically be programmed to die. So that's where we come into apoptosis. So apoptosis is, you've probably heard this term a lot, programmed cell death. Glossary term, remember that, super key. So in general, know that apoptosis, again, can be induced by different things, but it is very ordered, it's very programmed, and it's a very clean and efficient way of cells dying. It's a very necessary process as well. It's not always a bad thing. Sometimes it's necessary. It's used as a process as well to control for cell growth, so to make sure that there's not too many cells in the body. When you have old cells, to get rid of them, cells that aren't serving a function anymore, getting rid of them as well. But then also kind of what we've talked about, like if there is DNA damage that is irreparable, then we'll have apoptosis usually as well. So in general, again, I'm not going to go too in depth, but you have your intrinsic, so think about the term, and your extrinsic. So think of intrinsic coming from inside, so can cause by like cell damage or damage to DNA, things like that. And it's caused by basically this molecule from the mitochondria, so called cytochrome C. Again, probably not too key to remember all these details, but just understand that it comes from within the cell. And so once this little molecule is released, we've got all of this kind of thing, and it leads to apoptosis. On the other hand, with extrinsic, it comes from outside the cell. So we have this little molecule, and so this is the cell here, and it comes from externally basically, and it signals the cell again to program and have that apoptosis. Because that can be caused by extracellular things, such as infection, for example. As you can see here, again, you don't need to know the details of this, but if you see the term caspases, you can see that it's quite a similar process that results in ultimately the same thing. So while the initial thing may be different, the end result is the same. So we're looking at this idea of caspases. They are really important. I really highly recommend when you start to write your short answer questions and you're answering stuff with apoptosis, remember the term caspases. They're very central to this whole kind of process. Basically what they do is they initiate apoptosis by breaking everything down, like chopping everything up. So the cytoskeleton is what gives the cell its shape, its structure. So they chop that up. So we're getting this, like you can see here, this compared to this. We're losing our structure. We're cutting down our proteins, our organelles, things like that. So we're getting the cell shrinking, and what we see here, it starts to form blebs. So these little like offcuts, I don't know, like these little extensions of the cell. They basically break up, as we can see here, and then they get digested very nicely by our little phagocytes. So this stuff, again, I may be introducing some foreign words here. You'll learn about them, it'll make more sense. But ultimately we have the cell here. We lose all our structure, everything gets chopped up, we're forming these little blebs. Ultimately, these blebs actually branch off. So you can see how it's very neat. We're forming these little compartments that have all these like dead material in it really. And then our little phagocyte, which is a type of immune cell here, gathers them all up and gets rid of them. And so you can see it prevents inflammation. So when you have your cells dying, you don't know, right? Like you're not seeing this really gruesome process where you're getting inflammation, where you're getting pain, anything like that. It's very neat. It's very efficient. So that's why it's that idea of programmed cell death. It's very programmed, very clinical almost. Okay, hopefully that makes sense. I know that was a lot of terms, so hopefully that's all okay. But again, pop anything in the Q&A if you have to. So here we have a ranking poll. So move the phases around so you can rank the steps of mitosis in the correct order. But yeah, hopefully that's making sense. Any questions, pop them in the Q&A and we'll get to them. Perfect, give you a couple of seconds more. Okay, lovely. That's looking pretty good. I know the answering system is a little bit different, so hopefully yours looks like what I've got up on the screen here. But remember that acronym. Acronyms, other memory aids, mnemonics, whatever it is, really, really helpful for bio, especially when there's a lot of content. I think I saw a question about study tips and stuff like that. Yeah, try to think about little memory aids that can help. And again, trying to make connections when you can. And it can really help get things in your brain. So with processes, when you have a lot of steps, yeah, remembering acronyms and stuff like that is really important. So PMAT, prophase, metaphase, anaphase, telophase. And obviously try not to, I mean, you can rote learn them, but try to think, M for middle or A for apart, stuff like that. That can help as well. But just remember the order and obviously what is involved with them. Okay, I think that was it for the cell cycle stuff. Oh, okay, never mind. Don't worry, we've got a little bit more. So this is when we're thinking about, okay, so what we were talking about before, this is kind of all the neat stuff, as I mentioned. So when stuff goes right, when stuff goes wrong. So when something goes wrong in the cell cycle and you have apoptosis, that's exactly what you want to happen. The cell dies, everything's good, nothing kind of negative is going on. However, sometimes we can get issues with the process of mitosis, which we discussed before, and it can lead to these things called neoplasms, which are basically just these unwanted growths, you could say. So they can be described as benign, potentially malignant, or malignant. So this is when we're starting to get into things such as cancer. So benign basically just means not harmful in this kind of context. So we're thinking of masses that are localized. So they're just sitting there. The excess mitosis, it's not kind of branching out. It's not affecting anything else, any major organs, anything like that. It's benign, right? It's pretty limited to where it is right now. Potentially malignant, that's kind of our step upwards. So that's when we're thinking about, at the moment, it is relatively benign. It's still localized, but it has the capacity to invade other tissues and become cancer. So that's why potentially malignant. Once it is malignant, that's what we're thinking of tissue invasion. So we have this excess mitosis and it's becoming harmful. It's basically spreading into areas where we don't want it to be. Okay, so in terms of this idea of what can lead to malignancy and even metastasis as well regarding cancer, we have these things called oncogenes and tumor suppressor genes. Again, I'll try to cover these as basically as I can. Don't be alarmed if you're not grasping it. Again, there is more to it and it'll probably make sense once you learn it in its completion. But oncogenes, basically, they have to be activated, whereas our tumor suppressor genes, as you can see here, like the arrow kind of means like, yes, like activate it. And this little thing means block it. So maybe I'll talk about our tumor suppressor genes first. So our tumor suppressors, as the name suggests, they suppress tumors. So you may have heard this one called P53. It's probably the most like famous talked about one, whatever it is, because it's the one that is often dodgy when we think about cancer. So P53, its role is, when we looked at that checkpoint before, if you remember that diagram, that'll suppress tumors by when there is kind of damage to the cell cycle. It's important in stopping it. So when we've got issues with mitosis or when we've got issues with DNA damage, cell growth, whatever it may be, the tumor suppressor genes job, or the tumor suppressor's job is to stop the cell, right? It's to say, okay, something's gone wrong. Let's stop the cell. Let's, maybe make it to apoptosis. Let's try and fix it, whatever it is. With certain cancers, we see that the tumor suppressor, such as P53, it basically becomes faulty so that when there is an issue with the cell, we don't have anything saying stop it. So you've got DNA damage or you've got an issue with the growth of the cell. There's nothing there saying, okay, let's pause, let's stop the cell from growing. And that DNA damage or whatever it is that's gone wrong, that continues to be in that cell, and that cell will divide. And then that's when you've got issues, because then maybe you've got excess mitosis or you've got a mutation that can lead to cancer. On the other hand, we can also have our oncogenes. So our oncogenes are basically these genes that are capable of leading to cancer ultimately, and you have what are called proto-oncogenes. Again, I don't want to confuse you too much, but proto-oncogenes, they are these genes that just hang around, and to basically be negative and to lead to cancer, they have to be activated and become oncogenes. If you think of like oncologist, it's someone that specializes in cancer, right? So an oncogene, it's kind of this cancer gene. So if you have a proto-oncogene, it's not doing anything, but once that proto-oncogene, perhaps something is mutated in that gene, something goes wrong, and that gene is now activated, it becomes an oncogene, and that can lead to cancer. So can you see why there's like pluses here and minuses here? Because when a tumor suppressor gene stops working, so we've got it kind of being knocked out, that's when we're leading to cancer. And here, when we get our oncogenes activated, that's when they can lead to cancer. So the way that I think of it is tumor suppressors I think of as the brakes, oncogenes I think of as the accelerators for like a cancer car, if you kind of think about it. So your brakes aren't working, that car is going to drive straight forward. You're pushing the accelerator with your oncogenes, the car is going to drive straight forward, and that can lead to cancer. Some of the traits we're thinking of is uncontrolled cell growth. So we're just thinking of heaps and heaps and heaps of mitosis that cannot be stopped, basically. And that apoptosis that we really would desperately need in a situation, it's not happening. And then that's when we can get other things such as cells invading tissues, organs, spreading of cancer through the bloodstream, stuff like that. Okay, in terms of mutagens, we'll talk about them in the next slide, but mutagens are some of the things you would have seen this in a lot of health campaigns. I'm sure you guys will be aware of some of them. So if we're thinking about UV radiation, why it's important to wear sunscreen, when we think of like melanoma, stuff like that. Cigarette smoke, so obviously we've heard that a lot in like lung cancer, things like that. Other things such as, processed meats, when you barbecue something and it goes like really black, that can be carcinogenic as well. And certain infectious agents, so we've got HPV, H. pylori as well. They can cause infections that can lead to cancer too. Okay, so in terms of the actual definition of our mutagens, so basically factors that cause mutations in DNA. Again, think of the words mutagens, so they're leading to mutations. Mitogens lead to mitosis, so just be aware of that. With bio, it's not a huge thing to get your spelling wrong, especially because you guys are in 1-2, your teacher is the one who'll be marking it, so I'm sure they'll be relatively lenient. With VCAR, like once you get into 3-4 and your exams, the rule is that you can make spelling mistakes, it's fine, as long as they're not basically other biological terms. So if you were to write mutagens and you wrote mitogens, like even though you're thinking of mutagens, because mitogens are an actual biological term, that spelling mistake wouldn't be accepted. So hopefully that makes sense. Like if you were to write like mutagens with 2 T's, like they know what you meant, you know what I mean? Like mutagens aren't another term. So just be careful with things like this, like mitogens and mutagens, they sound the same. They're like within the same realm of like cell division and stuff like that. So just be really careful. Carcinogens, basically what we were talking about before, those tumor suppressor genes, those proto-oncogenes, they're kind of more directly affecting them. And so that's why they're really leading to that cancer. So obviously mutagens, stuff we talked about before, they cause mutations in DNA, and those mutations can lead to cancer. But sometimes when we're thinking of carcinogens, those are like specific mutagens that cause cancer. Hopefully that kind of makes sense. So just some of the stuff we talked about before in terms of chemical things, physical things, biological things as well. Again, I'm sure these examples, you guys would be able to think of some of them off the top of your head based on like public health campaigns and stuff like that. Okay, so there are some other factors that may lead to, or I guess, aid the process of cancer developing. So loss of immunity. So if you are an immunocompromised individual, so basically your immune system um isn't working as well as it should be, then it's not as strong perhaps. Perhaps the cells, there aren't as many immune cells as there should be. This may be more likely to lead to cancer because in the event that perhaps these neoplasms, you have these unwanted growths, you have these genes kind of going on, your body may be able to destroy them. However, if your immune system is weaker, it will probably be less likely to. So that's why we can see that as well. And these are just some general things to be aware of. Again, I'm not going to go into them in detail, but think of metastasis like spreading, evading growth suppressors. So where, evading apoptosis as well. So you want to kill the cell. Cancer cells can be very tricky, and they can basically escape the process of apoptosis, and you can't turn off the excessive mitosis basically as well. So that's what we can kind of see here. Okay, so we're on our last bit of cell stuff now. So looking at stem cells, again, you guys may be familiar with this, but stem cells are basically like cells that are a blank canvas. So they haven't differentiated into a specialized cell yet. So my teacher used to say like, I don't know, you can kind of think of your cells as stem cells, haven't specialized into a uni course or a TAFE course or, whatever vocation, stuff like that. And you might think of like babies as really basic stem cells. Then you go into primary school, maybe you're a little bit more specialized. Then you go into high school, you're choosing a VCE subjects, you're a little bit more specialized. You go to whatever you do after uni, then you go into a job, after school, then whatever you do to get to a job. And then by the time you're an old person, you're very specialized. So I don't know. That's how I sometimes think of stem cells as well. Basically, this idea of they come a little bit more specialized and a bit more specialized and a bit more specialized. So we can see that here. So obviously these are like all examples of different cell types, and they all come from the same basic one. So if we're thinking of in an embryo, so thinking of like the really basic like fertilizations just kind of happened. Obviously the cells that you have in there can become anything. You then have some other cells that are pluripotent. So you have totipotent or totipotent. I don't know, you hear it called some in different ways. But basically it can become anything. So I think of like toti, think of like total. Pluripotent basically means it can become most stuff. So we have here like endoderm, mesoderm, ectoderm. So these are kind of technical terms, but basically when you're in the like as an embryo, there are these three different types. And the reason why these are pluripotent and not totipotent, totipotent, they can become ultimately anything, but these cells cannot become like each other or that basic embryonic stem cell. So that's why it's not totipotent, if that makes sense. But they basically can become anything. Then they become a little bit more specialized, and then we call them multipotent. So you can still become some certain types of cells, but just not everything. So if you're thinking about bone marrow, skin, cord blood, whatever, stuff like that, they can become different things. So for example, bone marrow, so you can become a red blood cell, you can become a heart muscle, so like a cardiomyocyte basically. You can become different cells, but see how they're within kind of a similar realm. But these cells can't become a lung cell or they can't become a skin cell. So that's why we're a little bit more specialized. And once we are specialized cells, so once we're a red blood cell, we're not becoming anything else. So you're completely specialized there. And that's what we'd call unipotent. So think of uni as one. You can only be one type of cell. So like a skin cell or a neuron, you're only dividing to become that exact same type of cell. You're not dividing to become a different type of cell. Hopefully that makes sense. You can just see that kind of level of specialization. So hopefully that analogy of like a primary school child is kind of making sense. Okay, in general, we can see here our embryonic stem cells. So remember this idea of endoderm, mesoderm, ectoderm. They're just parts of the embryo really that can give rise to different things. So you can see how they're totipotent or pluripotent. So they can become a lot of different things, and they can replicate indefinitely. So basically they can replicate and replicate and replicate and replicate. So if you were to, I guess, like harvest those stem cells, they can become anything and they can replicate continuously. If you were to get an adult stem cell, they are multipotent or unipotent. So thinking a little bit down the line, you're a little bit more specialized, yeah? So you can use these stem cells to repair damaged tissue, stuff like that. However, these cells cannot replicate indefinitely. They still have a finite time because they are from an adult stem cell. That is the basic gist of it. Obviously, there are ethical things associated with this. So you can see that there's pros and cons of using embryonic stem cells and adult stem cells, but questions are raised when it comes into, harvesting embryonic stem cells or harvesting adult stem cells as well, kind of reversing them, things like that. But yeah, that's kind of a basic gist of stem cells.